Assalamu alaikum. Today we are going to talk about the second week of embryogenesis. Here again, this is the blastocele, and this is the inner cell mass or the embryoblast, the outer cell mass or the trophoblast. This is the zona pellucida. Here the stages of development starts by fertilized ovum, which is the zygote. Once start division by cleavage, it gives the morula, reaches the endometrium, which is the internal lining of the uterus. It's named the blastocyst. It gives two structures, the trophoblast and embryoblast. Here again, this figure shows the blastocyst. This is the trophoblast, in the inner cell mass, the embryoblast, surrounded by the zona pellucida. Second week of embryogenesis is often referred to as the week of twos. This is because during this week, when the appearance of the embryoblast, extraembryonic mesoderm, and trophoblast each separate into two distinct layers. Additionally, there are two cavities that develop within the embryonic unit at this time. These are the structures that appear during the second week. That's why it's named the week of twos. Here is the embryoblast giving two layers. This is the amniotic cavity, and this is the yolk cavity, so we have two cavities. And the trophoblast here, this blue color, is divided into cytotrophoblast and syncytiotrophoblast. And then lastly, the extraembryonic mesoderm. This, you can see this dark color, it participates in supporting the epithelium of the amnion and the yolk sac, and also it has a function in the development of the fetal blood. Here again, the embryoblast is divided into hypoblast and epiblast. Cavities, we have amniotic and primitive yolk sac. The trophoblast gives the cytotrophoblast and syncytiotrophoblast. That's why it's named the week of twos. At this stage, the embryo is named the bilaminar germ disc, since it has two layers, the epiblast and the hypoblast. These two layers, or the embryoblast, is located between the amniotic cavity and the yolk sac. At this stage, the implantation process starts, in which a developing embryo moving as a blastocyst through the uterus, it reaches the uterus, and it makes contact with the uterine wall and remains attached to it until birth. The lining of the uterus, the endometrium, prepares for the developing blastocyst to attach it by or via the many internal changes. The implanted embryo becomes more deeply embedded in the endometrium. Then, further development of the trophoblast, it will give placenta. The development of bilaminar embryo, amniotic cavity, yolk sac also occur during this time. Here, this figure shows that this is the embryo. It is implanted in the wall of the uterus. This is the endometrium. As I mentioned, the inner cell mass of the blastocyst divides into epiblast and hypoblast. The epiblast contributes to forming of the overlying amniotic membrane and amniotic cavity. The hypoblast contributes to the formation of underlying yolk sac. Again, the trophoblast, which represents the outer cell mass of the blastocyst, it differentiates and invades maternal tissues. It gives cytotrophoblast and syncytiotrophoblast. Maternal capillaries, trophoblastic lacuna, filled with maternal blood will be seen at this time, and the embryo is joined to the trophoblastic shell by the connecting stalk, which will develop later on to umbilical cord. It attaches the placenta to the embryo. This figure shows the embryo here, bilaminar germ disc, because it has two layers, the epiblast and the hypoblast, overlaid by the amniotic cavity, underlaid by the yolk sac, surrounded by this layer, which is the trophoblast layer, first division, the cytotrophoblast, and this green color represents the second division of the trophoblast, which is the syncytiotrophoblast. As you can see, it's embedded inside the wall of the uterus, Here you can see these spots represent blood that flows from the maternal circulation, will later on join the lacuna that will be formed here, to be filled with the blood. So the circulation will start. Here, these are the lacuna that appears in the interstitial trophoblast. The blood comes from the maternal circulation to settle down in this lacuna. Here you can see almost the blood is filling this lacuna and the interstitial trophoblast. As you can see, so this will supply the developing embryo by nutrition and oxygen. Further development, you can see that almost all the lacunae are filled with blood, and here this is named the utero-placenta circulation. This begins to act so to provide the developing embryo by oxygen and nutrition. This figure shows or represents the flat ovoid shape bilaminar germ disc. You can see it is the upper layer, it is the epiblast, and the lower layer, which is the hypoblast. This is the hypoblast layer, a localized area. Certain area of this hypoblast, the cells will change into columnar shape, forming a structure named the precorder plate. We'll talk about it later. This is a histological feature of the embryo inside the uterus. This is the embryo. This is the connecting stalk that binds this developing embryo with the wall of the uterus. Implantation sites usually are either posterior or anterior to the anterior or posterior wall of the uterus. Sometimes abnormal implantation sites appear. This is called ectopic pregnancy, maybe abdominal wall, mesenteries, or ampullar region of the uterine tube.